Okay, great. So I've been given this job to talk about vector and pathogen genomics. Uh, Last week when we had Chitra with us, she talked in really great detail about the pathogen genomics. She went through a lot of techniques, using NGS, pipelines, uh, different resolution. I hope you all remember all of that, because I'm not going to touch upon that at all. So, but before I actually even begin to discuss anything, you guys understand really roughly, right, what is genomics, right? Right? I don't need to go into the detail of that, right? But why do you think it's important for us to worry about any of these vectors? When I say vector, this is not mathematical vector as in something which transmits the disease, okay? We are all on the same page. Why do we study vector genomics? Mosquitoes are everywhere, ticks are there, they're there. That's good enough for us to know there is a risk, right? Kavita gave very nice overview of that. So why should I worry, spend so much money and look into their genomes? Potential to adapt, okay? And anybody else? How do you do that? How, sorry, could you please explain that again?
Now, if you find the variations in the genomic sequence, there we can understand the frequency of changes, in what direction it favors the organism to adapt better.
So it's adaptation basically you're talking.
Yeah.
Right.
Finally,
Yeah.
the adaptation only.
Anybody else? I'm still not listening the buzzwords, which I am actually hoping for. Why should I worry about that? To see what stresses them and how vectors behavior change. That will, I will infer that from the genome. Vector? Okay, immune system. Come on, guys. Why do I worry? If you ask me to do genomes and genomes of some Anopheles, Culex, or whatever, why should I be doing that? Huh? It's so much money and effort, plus computational headache, Tell me, Ishwarya, you've been working on KFD. Have you done your ticks genome? Close to what? Me?
So if you could understand genomics of mosquitoes, for example, we could understand why they are developing resistance to insecticides and
Very good.
everything.
Now people are waking up, okay? Resistance to pesticides. Anything else? Anybody? Come on. We haven't even touched the main thing yet.
If we can check for some conserved regions across the genome that contributes for the pathogenicity or
Okay, yeah, that's fine.
that's fine.
Yeah.
But you haven't written that on board, which means it.
Okay. No. Once you have the genome, you can look for anything. But my question is, people talked in great detail about vector and everything. Once you know this is something which is transmitting the disease, looking at the way people were also looking into, you also had some whole GIS thing yesterday. People talked about the home range of, I don't know, tigers, elephants, and everything. Would you not worry about
To identify the origin of evolution,
origin of evolution?
origin of point from which it has been acquired potency of transmission of the disease.
Okay, origin.
Sometimes, early of its existence, there is somewhere where the sequence is different. Then, meantime,
So basically, you're talking about population, how different the populations are, right? Population differentiation, yeah.
Yeah, you can tell over an evolutionary period which kind of lineages have been successful
Right.
in spreading
So
disease.
there is population differentiation, and then there is evolutionary history. Okay, so I still haven't heard one more thing. Then I'll move on to my next slide.
We can also use this
Huh?
sequence as for prediction from where that pathogen or the vector moved from one place to the other.
Yeah, so that would be in polygraph. So basically, you're tracking the different places and everything. Anybody else? Co-evolution with what? I put such a beautiful slide here for you guys to give some inspiration, some ideas, here. People been talking about Hawaii, birds, malaria, everything. Nobody is saying anything here. Huh?
To identify or understand their dynamics and to take proper control measures. Dynamics.
Dynamics as in population dynamics or dynamics in their...
Transmission.
Why are you all hung up on transmission part here? I will squash a mosquito and I'll make the genome. How am I supposed to know whether it's transmitting anything or not? Because I'm looking into mosquito genes right now.
So understand spread
Spread of what?
of the vector and pathogen both.
Spread of?
Both the mosquitoes as well as pathogen.
Yeah,
across space.
so when somebody's talked about the spread of the mosquito going from where to where, actually, once you have the genome, you're looking at population differentiation, then everything that I think falls into that. Population dynamics I will give you because it will come in some of these things. Sorry? Yes, that's the thing. No, vector control is a big deal, right? Once you know the genome, you know where the adaptation and everything going on. And there are a lot of things people are doing. For that, you need to understand the genome of any vector species. So, yes, but I just want to, I have put those things there, but I just want you guys to just look at this figure, which actually kind of tells you the evolution of how things have changed, where we were in terms of, looking at a small section of the genome where we were doing Sanger sequencing. Even now, people do Sanger sequencing, right? You kind of amplify mitochondrial, cytochrome oxidase, whatever. To people are doing DDRAD, which basically chops the different sections of the genome, and it's more of a shallow kind of sequencing. You are going towards this whole genome resequencing, or you can do whole genome sequencing, a very high resolution one. And it kind of tells you can analyze different kind of things based on the kind of data you have. So this is just to put things into perspective. Now, this also, Chitra actually just kind of ran through that day, that when you are sequencing a genome, so I'm talking about genomics, so I'm only talking about genome-wide section here. When you're sequencing a genome, depending on how much money you have and how many individuals you want to do, you got to, and also the questions you are actually interested in answering, you kind of decide that how much and what kind of sequencing I want to do. So you go for whole genome sequencing, which is actually going through the different sections of basically covering the whole genome, and also the depth of the sequencing is that to be like 5x, 2x, 1x, whatever. The deeper you go, the better kind of confidence you have in calling the data, what you have got. Are you all with me? So when you go for sequencing is the easiest part of the whole deal, by the way. Catching mosquito is difficult. So you get the animal, you get the vectors, and you get hundreds and millions of read. And then you align them against to the reference genome. Namrata is the person to talk about the whole pipeline. She can explain it to you, how that works. I'm not going to go into the detail of that. So you get the deal, right? So because I'll be going through these things here and there in some of the examples, which I will take later on. So now you have a very good sequenced data set, and this is how we kind of call alleles in our, or the SNPs. You know, this is the reference genome here. These are all the different reads. So you have this position, which is, most of them are at the C location. This is all homozygous. And then you can see this is a heterozygous, which is coming into different. So the larger the number of reads, the confidence you get in calling that as a homozygous and heterozygous. Now, this homozygous and heterozygous thing is very important, right? How homozygosity and heterozygosity work in a population. So is it a heterozygosity is very good or homozygosity very good? Can somebody quickly? Okay, so how? Sorry?
Success rate of continuation to the next generation is quite high in spite of communications in the environment.
Yeah. So say I take example from 3, yeah, please use the mic for that. So I'm taking 3 different sorts of, this is all in terms of the genotypes, not by the species here. This is one population. Then there is a population 2, where you take some individuals from here, and you have very representative sample. And then you filter out third population, where you basically end up with only one kind here, right? You see what's happening here, right? You're basically reducing your heterozygosity. So if I have SNPs or genomes for sequenced from these individuals, so the heterozygosity, say for example, for here, if this is heterozygosity, you'll say this is 0, this is say going up to 3, somewhere here is say is 1.5. So all the SNPs which different heterozygosity will be for a very diverse population would be very higher. Then you go for the next one. What do you think is going to happen here?
Less.
It will decrease?
Yeah.
How much decrease? Little? So will it be close to 0 also? No? Then? Little bit. But there will be mixed pattern going on. What about here? Close to?
0.
Close to 0. Very good. So this close to 0, when we are recording, this is something which is referred to as runs of homozygosity. So when you are looking into populations, say we see it a lot in the insectary populations for mosquito or different things when we breed. And this is something which actually Kavita also mentioned, that how that affects the fitness. We have seen in our insectary population, when you have a very wild population, their fitness, the number of eggs they lay, the wing size, it's kind of pretty good because the fitness is good. But as you reduce it to the one generation, next generation, say up to 16 generation, the egg count also reduces, but also their wing size also becomes very less. So you understand what I am going to talk about here. Right. So imagine if you take some population to landscape approach. Everybody must have read about Hardy-Weinberg in equilibrium, right? I'm not going to go details of any of the stats and formulas here. Can somebody explain it to me what that means and how that works? It will make my life easier going forward. Yes, sir.
Allele frequency.
Yeah.
The percentage of allele frequency goes to the next generation. That depends on the heterozygosity. If there is a problem arise to the one population, the success rate of the survival of that species, that depends upon its survivability to that question. If one problem posed by the nature, so there, out of so many allelic pools, only one allele, one type of allele will successfully select in that condition, where the remaining alleles will be eliminated. Meantime, if there is any mix of alleles, again, it increases the survivability. Again, that also depends upon the type of problem it is posed by an environment.
Okay, so you're explaining to me that there are disturbances, basically there are problems, but that's not what Hardy Weinberg equilibrium is saying here. Anybody else wants to attempt? Mike, you need.
In a Mendelian population,
Sorry?
when evolutionary pressures are absent, the allele frequencies remain the same throughout.
Constant, yeah, from one to another.
Yes.
And unless there are
external evolutionary pressures
like,
acting on it.
Like what?
Like genetic drift, migration, and gene blur.
Okay, so which is kind of, we see all the time happening, right? It's just a matter of that when you have the data, you need to understand which direction it is going and what might be playing or happening in that population. So people understand this bit as well? Now going back to this whole doing the genome sequencing versus the what we used to do, say even now people do microsatellites, which are the small tandem repeats. It's also on the quality or resolution of the data also. So for example, some of the studies people have done, they use say 13 micro, this is a shrimp by the way, so not a mosquito, where they use 13 microsatellites and they looked at the population structure. And they found there was hardly any resolution in the data. It's just kind of a one panmictic population based on the 13 microsatellites. But when they do the same analyses using these SNPs with the genome-wide SNPs, they see some kind of structure in the population. This is not written on stone. It changes with different dynamics and different population. But one of the reasons people are inclining towards using more genome-wide SNPs is this, that it kind of increases your power to analyze the data. And also, there are not macrosatellites available for lots of species. And trying to figure out microsatellites for all the species is kind of cumbersome as well. It's been recently done for Aedes aegypti as well. This is from some different populations where, again, they showed that when they did the SNP, they showed a very clear-cut demarcation in the population. This is in, I think, Vietnam and some of the Southeast Asia, as versus using the microsatellite, which just shows a very mixed kind of population structure. And also, it depends on the sample size, also what you are using and trying to do this kind of analyses. So with respect to mosquitoes, which Kavita actually asked you all that, what are the different mosquito-borne disease? Everybody piped up and said malaria, filaria, dengue, and chikungunya, and everything. So there are actually 3 key mosquito genera which are responsible, right? Anopheles, Culex, and Aedes, right? So it becomes very, very important that we must have genomes for at least these 3 species to start with, because it will allow you to understand how things are, which everybody is discussing here. So I just want to give an example for this particular Anopheles conundrum, which is called as a jumbled genomes here. So Anopheles is a very, very diverse species. Africa has very many, I'll talk about that in a bit. But in India, for example, we have Stephensii, we have Anopheles culicifacies, which all are kind of responsible for transmission of malaria. And I think that's one of the reasons you have so many species. It's hard to target which one to go for, and it changes with the regions and everything. But what I find very interesting about that, look at the divergence pattern which these species has diverged. For example, these Culex and Aedes aegypti, they have diverged somewhere here 150 million years ago from this main clade, which is diverging here from Anopheline at 100 million years ago. There's a massive difference in the genome size of these species. Aedes aegypti has a very big genome, something like 1.5 GB, as opposed to these Anopheles, which is something like 200, 54 MB or something. It kind of varies. And if you understand, if you remember the talk we had last week from the bacterial genomes when we were talking about those transposable elements and how the genes are moving within the thing, some of that actually applies to Aedes aegypti as well. The fact it has such a large genome, it does have a lot of transposable element, which allows this species to kind of invade and adapt to new locations very, very fast. That's actually one of the reasons. The second thing I think I find very interesting about Anopheles, which I think some of them people are asked that how fixed these vector species are on their host, right? When I showed the phylogeny of Anopheles versus Culex, and then you have other vector species going on. If you look at the genome of, say, Anopheles gambi here, Anopheles gambi has so many olfactory and gustatory receptors which are more prone for human blood and human sensory. And that's why this species is so anthropogenic and it likes humans basically, compared to any of the species which people have compared to rest of these two species which can go between different kind of habitats. So they did all these genomes for these different species of Anopheles. These are 16 genomes which was prepared. And one of the species which there are a lot of literature actually related to this also is Anopheles gambi. If you see this figure I have put here, until 1970s, it was considered as only one single species. This was a species complex. Before you actually go ahead and do the genomics for any species, what do you need to know about it?
Any species available, genome data available for the
I
closest?
forget all of that.
Okay.
I've written. Taxonomy. You need to know what you are crushing and actually sequencing first of all. And the problem with this gambi is morphologically nearly indistinguishable. There are so many these types which later on 1985 they had described here, but until 1970, there is no way on planet Earth you can tell this is a different form of or different species or subspecies of Anopheles gambi. Now, if you cannot distinguish it taxonomically, and if those species are dispersed in different kind of habitats, there is no way you will be able to understand why some of these things are how they are distributed. So for example, when they figured out that these species are distributed, say, forest, savanna, or Bumako or Bisu, whatever different kind of habitat, the reason when they did the genomics for this, they figured out there is a huge amount of inversion in their chromosomes. Are you familiar with this word of inversions in chromosomes? No? Okay. So inversion is kind of a polymorphism in a chromosome. So lots of species have, birds have. So for example, you have, say, these sections of genomes. Somewhere here, say, especially in these mosquitoes, they have these regions. They're kind of flipped. So mosquitoes have 3 pairs, so of which 2 are autosomes and 2 are sex chromosomes. So leave aside the sex chromosome, but in autosomes also, the sequence, the way these chromosomes are arranged is not the uniform across all of these species. And what it means that, say, if these species are distributed, say, in a savanna kind of habitat versus a desert region, and even if they come in contact with each other, these regions can combine, but these cannot. So they cannot hybridize so easily. So there will not be any viable progeny, if at all, which kind of makes this a kind of an exclusion boundary between both genetically but also habitat. And this is one of the reasons having these inversions cause these species to be fixed on these kind of habitat. Now imagine all of these pockets of, for example, here for Arabiensis, this is where it is found. And this part of is all occupied by this gambi over here. And this is the area where it is kind of found more into the desert kind of region, all because of the way they have chromosomes arranged. None of that was known unless they had these genomes available for these species. Same for all other species. So what a species is doing, they are distributed in space and time, but they also have evolved for these years to have these structures. And later on, until very recently, I'm not talking very far back here, they identified this Gambi as form of two forms, M and S, which M was actually called as Anopheles colusi. It's made after a very famous scientist who studies Anopheles Gambi for so long, all his life in South Africa, sorry, African continent, and Gambi in different parts of Africa. So once you know that they are two distinct species and they are different forms, it makes your life easier now, right? You can tell them apart. You can look at those chromosomal structure when you catch them. You even know that most of these are confined to females, not to males. Now, why is that important for a mosquito species? Can somebody tell me? So if the chromosome structure, the way they are aligning, more selection is on the female, not on the male, what would that will tell you? Why that's important to understand? Come on, people, wake up. We just had a cup of coffee. Guha mentioned in his talk that reproductive isolation in mosquitoes, right? And who is the biting all these for protein? Females. So which means there are some kind of unique structures or something going on in the female genome which males do not have, right? So this evolutionary point of view, this must have happened at some point in time. But for us to understand where and how those changes are happening makes female more important than males, basically. So a lot of studies when people are doing these genomics and everything, what they do is actually they collect the females. Actually, there's one more way to identify mosquitoes is looking at their polytene chromosome, which is there in the larval stage in the salivary glands. And as they transform to the adult life, it kind of disappears. But imagine extracting a salivary gland, polytene chromosome from a salivary gland. You need to have a really good expertise to do that. And it's just, I think very few people I know. One in our secretary, she does it. So it's a very impossible thing to do. But once you know the genomes and when you know these forms, I think, and these chromosomal structures, how they are arranged, I think it makes life easier, and then one can do that. So a lot of these studies are when they are looking at the genomics or structure, they are done using the female samples, basically. They collect the larvae, and then they rear them in the lab, and then they know definitely this is what, and then they go ahead for these different kind of analyses. Now, you have to, after this long history of this is pretty much my life. I was born in 1972. Took them so long to figure out what those species are. Can you tell me what could be done with this information? Malaria is a top 1 species in Africa. Malaria kills more people than COVID has in Africa. Remember that. So more than 1 million people every year. Remember, that's a big, big statistics. What else would you like to know about this species? If you know Gambi is causing this havoc, and there are so many of Anopheles subspecies are there in that continent. Huh? Yeah, but let's forget the transmission part. Just let's stay on the host part right now, which is the vector part, that we are looking at the genomes, we are understanding how species is there. They know that most of them are transmitting malaria, but not all of them. So, yeah. Mosquito is a very invasive species also. Is it not? Somebody actually used that word in last few days. It is a very highly invasive species. They're very adaptable, I was told just 5 minutes ago. They kind of colonize from one place to another place very much. You can track how they are going. If there is a high, say, pesticide use here, but they are colonizing different areas, you would like to know which populations are moving and what could be the possible route. What else would like to know, which will help you to going forward? Genetic diversity? What like from the ecology point of view? Okay, yes, very good. Yes, but I just showed you so many evolutionary processes. If you are looking into the genome of a species and you have all that information, you can figure out that how well established that species is. So from here to there, where it is coming from. Is there are signatures of drift? Is there a gene flow? What could be the factors that might be driving it? Or is it a very homozygous population? Is it a very because even in mosquitoes, it really does matter, right? If you are a very inbred population, maybe your vectorial potential will not be as high as for a mainland population. Questions like that. So in order to understand that invasion uh, potential or how big competent vector this can become, I think it's a very important thing. So once they knew that this is what these two species are, they actually went ahead and they're trying to understand how Anopheles gambiae has colonized, say, from mainland Africa to the nearby Indian Oceanic islands. So there are several samples, as I was just mentioned, that all the female samples they collected from, say, Mali, Cameroon, Zambia, Tanzania, and all of these Comoros, Indian Ocean islands, basically. They do not know when these mosquitoes have colonized there. They all know that there are outbreaks of malaria and the species is basically there. But now they understand that this is a gambiae, which is actually has colonized these islands. So they did something like, I think, 15x sequencing for this work, something just under 200 samples from different populations. And what they looked into this genetic variability, that how that changes from across all these different populations. So knowing what I just was explaining to you guys, that from population 1 to 2 to 3, how do you put that into perspective? That how do you think that genetic variability can change from, would it be higher on mainland or would it be higher on the islands where they have gone now, or?
Because as the major population, like the founding population would be more heterozygous, then would have more variation basically compared to a small bunch that might have been transferred to an island, and they have maintained a population and all of that, but they started off with like a small bunch from the main genetic pool.
That is correct. And this is exactly what they are showing here also, right? So they are basically looking at, the biallelic SNP across different loci, and they calculating this genetic variability, and they're showing that Mali, which is actually in the mainland, has a slightly higher than in Cameroon, and then after that, I think it's Zambia. And by the time it reaches this island Anhuan, which is a very small island, it kind of diminishes. Now, there are several scenarios where this can happen, that you have this kind of gradient of genetic variability. Can somebody try to think about that? What might be happening? This is assuming what?
Sorry.
So there's a very clear-cut gradient here, right? So under what scenario this might be happening?
So the initial population started from there, and over a long period of time, they have moved towards the south, and then at the end of it, they came to the island. But if you find like islands that are far apart from the last island, then you might find lesser variability there. So the gradient goes down as you move from Mali to there, if you think the population has started like that.
Right. So you're saying that geographically there is as the distance is increasing geographically, there is a differentiation is also basically increasing. But don't you think this is also assuming that sorry?
Because of variations.
Yeah.
Because of variations. Because of variations in the genome sequence, which also influence its behavioral changes and its also morphological changes, whenever the host shifted to one zone, one area, maybe those sequences are quite fit, they support.
This is based on SNPs, by the way.
Fitness.
Yeah.
So which is adapted very well for that zone. So then slowly, if the population is very high, then slowly they migrate. If they migrate to other region,
Yeah.
If some, you consider 1,000 insects.
No, but
Now, what I mean to say that,
yeah.
so those sequences, those variations are highly supportive in that zone.
Which
Whereas,
zone? This zone?
Yeah, yeah.
This, which is mainland.
Mainland,
Yeah.
okay.
Which is a source population.
Yeah.
Yeah.
If there is something disturbance, if other flies, they don't have that sequence, but still they have different sequence. If those insects are not fit enough to that region, they move from that place to the another place. So where that variation supported by other environment. So where that population is stabilized to that environment. If there are other variations in the same pool, if they are not fit enough to that environment, they move to the other region. That means overall conclusion is that some sequences are, some variations are highly supportive for a particular geographical environmental conditions which supports for the survival of that species better compared to any other variations.
Do you guys agree with that? Deepa, you're shaking your head but not saying anything.
Basically saying about survival of the fittest in that particular environment. So which species is not surviving in that particular environment, it's shifting to a new place. So basically a multiple colonization is happening in that place from
Multiple colonization to the same area or different locations?
different area.
Okay, so somebody just said that this doesn't tell you that this is the source, by the way. It just all it tells you that mainland and islands have different genetic variability. It doesn't tell you where the colonization has happened. Colonization could have happened from Cameroon also. There are so many mainland locations, right? Tanzania also is very close to these islands, what they are showing here, by the way. So you'd expect it's by the coast, so that could have happened. All it's showing that as you were going, population are colonizing, the genetic variability is low, which could be as a result of drift or whatever thing. The scenario I was talking that if you had multiple colonization happening, say from the mainland, like the populations are being supplemented every now and then there was a scenario that all these genetic variability could have been similar across, but that's not the scenario you are looking at. The islands have not been, it seems like the islands have been colonized, but there is not a continuous supplementation of these mosquito, larval pools or whatever the method way they are being colonized is happening. So their genetic variability is quite low than the mainland, basically.
Could it be also related to the populations of humans there? Because in the islands there are lesser number of humans, and then in the mainland
True, but they also had outbreaks of malaria also in these areas as well. Yeah, so that's why it's a very important species of concern for that as well. I'll move on to this point. I'll come back to it in a bit with a different example for this. So they also looked into the population structure, which also fits with that Mali and everything kind of, cluster together. So all the SNPs confined to Mali are here. Cameroon is kind of coming close to this, then Tanzania and Mohali and Enuhan. So what also it tells you that island populations are also very different from each other, right? They are not kind of mixing, except for the mainland, and these two populations are slightly close to each other because they do share a lot of genetic SNPs from here. They also did something called admixture analysis, which basically looks into the different genetic clusters in these populations. So each bar here basically is telling you a single sample here. So the number of colors actually coincides with all the different locations of populations they are telling you. But wherever you are seeing the mixing of colors, it tells you that's where you have admixture of different populations. The genetic transfer of material is basically happening here. So for example, Mali has a lot of red, which is coming from your Enuhan, basically, and the blue one, which is from the Cameroon. Then Zambia and Tanzania here. Grand Comoro is pretty much, I would say, on its own. It has two genetic clusters, and then you have Mohali and Enuhan. So looking at this, it does seem like that Mali could have been the source from where the Enuhan population they may have. But what else also might be going on here? You have so many clusters going on here. They could have been colonized from somewhere else, which they may not have sampled. Is that a possibility? Namrata? Yes? Am I right? Okay, so this is just I'm not going to the detail of this analysis, how and all of it, but I'm just what the idea of doing this or showing you these is to make you understand that in terms of the invasion biology, the genomes can help you to understand how the populations are moving, and because vectors are taxonomically, morphologically are very difficult to identify, but also still you need to know. Yes, please.
Is that what we have discussed earlier is the same because of that reason, because of continuous mutation and selection, or because of sexual recombination, this one?
So this is just it's basically the way this analyses work is that so each sample is one sample. It basically the analysis tells you how many genetic clusters you have in a population. They could have been only 2, they could have been 4, they could have been more than 10, whatever. But when this analysis it tells you how many genetic clusters. So for example, this one sample has only 1, followed by the 2, which actually matches with the half of this and half of with the Cameroon population. So it just shows you that how the population are mixing.
Is that those two populations are mixing with each other?
Yes.
And also they are involved in the sexual reproduction?
Yeah. The gene flow is there, basically. There is a gene flow between those populations.
So, on the other way around, so another possibility of selection of the same species within the same region, regional selection.
If you have that,
that is also another possibility, or migration and gene flow.
If there is no gene flow, then you would have had the situation.
Both are true, or only one is correct, or another?
So the fact you do not have much mixing with the mainland population, say from here and here, it seems like those populations are very isolated.
Conserved.
Yeah, very conserved, because they are so kind of geographically far from here. And of course, mainland population are, say Cameroon and Mali are close to each other. So that's why you are expecting that to happen. Okay? Now, so this is for the Anopheles gambi. We, in Indian context, I remember yesterday it was mentioned that Nipah virus is in WHO list, is like the topmost virus, and also what other viruses? I think all the viruses people talk about. Anopheles stephensi is in the top list of mosquito in WHO. It's the most highly invasive species, and you can see we are in the hotbed of this species. We have pretty much all the native population for this. And in the recent past, when I say recent past, I'm talking about all these dates which I mentioned here. This mosquito has colonized a lot of countries, say in say Saudi Arabia, in Egypt, Ethiopia, this Horn of Africa. There have been very unseasonal outbreaks of malaria. The reason this species is so important is that you have only falciparum in the mainland Africa. But by having this stephensi, which can transmit both falciparum and vivax both, which is very dominant in India, by the way, we have both Plasmodium vivax and falciparum. This is why everybody is very worried in the south, from the African continent, that even if you remember, they just introduced this vaccine for malaria in Africa, which is actually only for the falciparum, not for the vivax. So imagine now you have a new species which is coming. It can actually derail all your plan for malaria-free Africa, basically. So it's a huge problem. Now, if you look at this species, this also has colonized Sri Lanka very recently and in Lakshadweep Islands. Now, the one thing about mosquito colonization, how the species are colonizing and moving about, people know that a species has arrived when nobody's doing a surveillance, by the way, unless you have a very good program going on, and some countries have seaport and airport, they kind of monitoring they do because of the yellow fever and other kind of things. France does very regularly because of all the change of climate change and for the Zika and other things. Many people do not know a species has arrived unless the population has established and you see outbreaks of certain disease, and then they start looking for that, why suddenly we are having these cases, and then they realize, oh, actually, we have an established population of, say, Aedes aegypti or albopictus in our region, and that might be driving these cases. And that's exactly what happened here in Africa, that they have these outbreaks of malaria, but when they noticed that there was some unseasonal outbreaks, they realize that why suddenly we are having, and they realize it's because of the Anopheles stephensii. And what it tells you that this species is quite robust. It has gone there and it has become very established, basically. So now there is a kind of a big race to figure out where are the routes from where these mosquitoes are coming and what could be the possible thing. We are basically interested in more from the Indian, in the Indian context, because if you see the whole of our India, the species were very prevalent in the north, but very recently it has come to the southern India in this region, and then to Sri Lanka and also in Lakshadweep Island. Just to understand why and how this species is expanding, so it's a freshwater species. It doesn't breed like Aedes aegypti, which can breed everywhere, in the small plastic containers. It requires fresh water. Usually people, when they have wells, cement tanks, or where they hold water, that's where are the main grounds for this species. But also, we have spotted in our Bangalore, for example, some of the tires and where you have fresh water, they do co-breed with the Aedes aegypti. So this kind of a coexistence is happening now. So that's a big thing. So we wanted to understand what does that mean from the Lakshadweep perspective. So Lakshadweep actually doesn't have any malaria case reported, ever, even though the stephensii is there. And there was one case reported some time back, which was also a imported case from the mainland. And there are some surveys people have done, as I have listed here. So you have lymphatic filariasis was mentioned. So lymphatic filariasis is from the Culex mosquitoes you have. You have albopictus, which there have been chikungunya outbreaks also have been reported from there. So we did this survey. This was just before the COVID came in the Lakshadweep Islands. And basically the idea was to document the mosquito ecology and diversity, basically what's there on this different 10 inhabited islands of these Lakshadweep. And taking it beyond just from the ecology, that if we can put a layer of population genomics also to understand where it came from. Of course, it must have come from India. That's an assumption, right? Because it's close to us and most of the human presence is there from the mainland India. But what about Sri Lanka? How did it get there? Do you know one thing? Sri Lanka is malaria-free. They declared themselves malaria-free a few years ago. So if you have a stephensii there, you're not malaria-free anymore. That threat is pretty much there, and the species is co-adapting to, say, brackish water. And so there are all kinds of things going on, which means having a genome of this species is very important, which we have. TIGS actually made one very nice genome a few years back. It's published. And that actually allows you to understand how the different regions might be evolving, or resistance and everything. So we did this survey. We documented all the diversity of the mosquito, and albopictus is the dominant species, as you can see, but stephensii was in very small number in very few islands. And when you see all these different islands, all the red dots are actually albopictus, all of them. We were really, really surprised that Aegypti was in such a minuscule amount. Aegypti actually is a more urban dengue mosquito. So in Bangalore, when you do the same survey, it's usually the Aegypti. Albopictus is in the back seat. But because these islands are more green, you have albopictus is in very. And stephensii, we found out of 10 islands was only in the 8 islands. So I'll just give you an idea. So if we got, say, 2,000 samples of albopictus, we have only 70 samples of stephensii. That's the number you have. We're having very low kind of numbers over there. And there's a huge dengue threat also there because of this huge albopictus. So if they have had these chikungunya or any dengue outbreak, albopictus might be driving that. Okay, wake up now. I'm going to ask a question. So you saw what I showed you, the plot for the Gambi, where mainland to island comparison I just showed you. If I do this similar analyses with respect to, say, all these ports I have marked here, Kochi and, say, Mangalore, and all these islands, and assuming, because these are the sea routes where people are taking the boats to these islands, what do you think might be happening to the genetic variability? A, with respect to stephensii, considering the numbers you know. B, with respect to, say, Aedes mosquito. which I have just told you the numbers. Will it be? Why? Reverse for what? Reverse with respect between Stephens I and albopictus? Okay. So the question I'm asking here is that we did this survey. We have, say, mainland samples from, say, 2 locations here, and then you have all the island samples. I just told you that what was the number of samples we have got from Stephens I, which just tells you that they are there, but they're in less number. So what kind of genetic variability do you expect to find in Stephens I, considering Aedes albopictus, which is so dominant on that island, and if I compare that to the mainland population?
Are less in this thing, so maybe they are more homozygous.
I'm talking about genetic variability just from mosquito point of view, forget the malaria. Okay. Think about the ecology of the species, how things, I explained that albopictus can breed in lots of places compared to Stephens I, still very specific. Saline water, I never mentioned that. No, I have not signed with them.
I think water availability is there.
On Lakshadweep? Yeah, so all the 10 islands are inhabited.
They are bringing the water from here to there.
Who?
For drinking water and...
No, they have a lot of water. They have, actually, the houses have their own wells where they keep the fresh water. And think from the ecology point of view, the numbers kind of I'm talking here, some idea what might be happening. For Gambia, you saw the whole mainland to gradient there for the island. Do you expect the same for Stephens I versus, say, for Aedes mosquito? Will that be similar situation or something different? Something. That's all I'm asking. Between? Less variation to what? So you have, say you have 2 mainland population here, population 1, population 2, these are mainland, and then rest are island, all the 10 islands of Lakshadweep. How do you expect the genetic variability for, say, Anopheles Stephens I? And then same for the Aedes. I'm talking about albopictus because Egypti was very less. Yeah. So you're saying here the mainland will have high genetic variability in for Stephens I, and then it will go down as we go down. What about here for albopictus? Sorry? So we are assuming that they have colonized from the mainland, right? Where will they come on the island? They could be from anywhere, any source.
No, I'm asking, is it linked to the Gondwanan movement of India continent from there or something?
That I don't know how long they have been there. I have not looked into their genome. I have not marked it.
This all island...
No. So don't go into the tectonic movement. I am talking about that you have these islands. They are reporting these disease. They also are reporting you that when and how they found the different vector species. Considering you have these islands and these mosquito species are dispersed around, one is in a very high number, other one is in a very few number. How do you think that will reflect in their genetic variability with respect to the mainland population? So one thing, I think you might expect that mainland will have a high genetic variability, right? Because that's kind of, right? But yeah, the question is for the islands.
It will decrease, it should decrease for both theoretically, but relative to Stephens I, it should not be as steep.
It should not be steep for Stephens I? Why is that?
No, it should not, for albopictus, it won't be as steep as Stephens I.
So you think it might be just like that?
Yeah, less steep.
But for Stephens I?
It should decrease over a period of time, but it will be steeper.
So I don't know what period of time. We have gone only once, right? And we are selecting these 70 samples from there. So
Yeah, so this.
It will be, you're saying it will decrease basically?
Yeah.
Yeah? So, okay, I won't make it this steep, but I'll just keep it there. Fine. Okay, so this is something I asked earlier also, that under what scenarios do you guys think this genetic variability can increase in a population? You're expecting people to go only once and say that some person in a boat went and dropped by mistake some larva. Some aircraft came and brought some Aedes mosquito and left it there.
Yeah, migration and mutation, I guess.
Yeah? Yes, yes, yeah. So I just wanted to say that there are only, so for example, Kavaratti is the one island which is connected, has an airport. And so you have direct ferries coming from Kochi and Mangalore also coming there. But within the islands, the movement is only by boat, by the people. There is no airport as such. So there is a lot of boat movement going on. These are not isolated, there is a proper ferry route and everything there. Where? In the islands? With respect to mainland?
To mainland.
In which species?
Species, I don't.
No, I'm talking about two species, Stephensii and this is Albopictus. I'm just talking about genetic variability. I'm still stuck on that.
Stephensii has a very restricted here and there kind of distribution. Hence, even if there is movement between islands, it is through ferries or boats, and there is water on ferries and boats. So there may not be that amount of movement, but whereas Albopictus has high amount of prevalence or presence, so there is common movement and a chance of moving here and there is more. So it shows a higher variation, but overall variation when compared to the mainland may be low. Like there is a separate cluster of variations in Lakshadweep, and there are like sub-clusters A, B, C, and they are showing inter-cluster variations. Whereas in...
so you're saying it will be more enhanced in Albopictus than
in
compared to
Stephensii.
Stephensii. Do you guys agree with that? No, everybody's waiting for lunch, basically. Okay, I mean, I don't know the answer for this, to be honest. But I don't know. I'm just giving you a scenario. This is why I'm saying, think. No, I don't even have fitness data for that. All I am looking at these genomes that we are sequencing and what kind of scenarios. Think from the genetic drift point of view, think from the isolation from the mainland, think about other processes which might be going, that how people might be moving, and are we talking about only one introduction? Can there be multiple introduction? If you have only, say, few introductions happening in a site, the genetic variability will be low compared, right? It's just population. Assuming that population got there and established, as opposed to you have people coming every time and bringing tons of boats and mosquito larvae and supplementing. But one more thing happens on island, which you guys must understand. My islands are very prone to catastrophic events, right? Cyclone. So many cyclones come there. So what if I do this survey just after the cyclone? And you have suddenly some of the mosquitoes species have become very less. So I would say this is what it is, right? There are no Stephensii, there are only Albopictus. Because we are going only one time. You can't live there. You're doing this survey for 4 months. These are the things. I am trying to bridge not just genome part here, but also whatever we have learned in terms of the community ecology, but also the species richness and aspect, that how do you actually verify some of those answers when you're looking at your genome data as well, which helps you to understand. So your answer can be correct, which makes sense to me. I don't know about other people, but it may be wrong because, as I said, we have few samples. Some of the things may change, may be dependent on that. But I'll show some data, but not all, because it's all very preliminary. It's not perfect. It's not like something written on stone. It will change by the time Namrata will publish it. So we recorded these species. There are so many of them. And it's a very high dengue risk there based on these incidents. So we did, everybody talked about island biogeography theory. So as the island area is increasing, the mosquito species richness is also increasing on these islands. which makes sense, which a lot of people talked about from different perspectives. I just wanted to show for the mosquito. But we also have been recorded the habitat for different mosquito species based on different pH and temperature. This is all actually based on the model, what drives the abundance of the different species. So for example, this Malaya gianirostris it only found in the natural habitat. It was not found in the artificial. When I say artificial, it's like man-made habitat, as opposed to Albopictus, which is there in both kind of habitat. And then you have these Egypti, which is in there, and in Culex as well. Okay. When you look at the community similarity, this is the richness or the distance in between the species with respect to the geographic distance. As the geographic distance is increasing in these islands, the similarities on the species is also kind of increasing. And that's usually is because of the Albopictus is so high. This is all driven by Albopictus in that population. And we looked into the coexistence mechanism. Now, going back to the genomics aspect of that, we did some 15x sequencing of something like 170 samples we have from all these populations, some Sri Lanka, some Bangalore, Kochi, and Lakshadweep. This is a very preliminary data set I'm showing you here. And we try to understand that how the genetic variability. What are you guys looking at here now? What do you think? Is it correct? Is it fits with your expectation, what you were just saying? And if not, then what do you think might be happening? These are all island population. These are all mainland population. Come on, we have only 40 minutes left. This is just the genetic variability I have plotted for each population here, just comparing. The way I was showing it for Anopheles gambiae, this is for Anopheles stephensii. These are all mainland population that we have done the sequencing for, and these are all the Lakshadweep populations. What does it tell you? Do you see a gradient here? As you saw in gambiae, that when you move away from the mainland, you don't see that in stephensii at all, right? Except for Sri Lanka. What does that tell you? Type of what?
Maybe they have migrated to these islands very recently.
Yes, Sri Lanka is the most recent colonized population compared to any of the Lakshadweep population. But what is very surprising about the Lakshadweep island population, based on whatever information I'm showing you, they all have pretty much the similar kind of variability as the mainland.
Lakshadweep condition is a very narrow condition which supports that population, where only the sequence which supports that narrow environmental conditions which fit there.
True, but just now he was saying that they are very patchily distributed, right? Which restrict them from moving and everything. But that apparently is not affecting their genetic variability, which also makes me think that the other point which I was trying to say, that these are islands. They are not colonized only once. They must have been colonized several times. People are moving constantly with boats and whatever. So they are kind of bringing these larvae, they're moving. And for mosquitoes to establish, say in different, even if somebody has, each house has a well there where you can actually find these stephensii larvae, but they do some kind of biocontrol also in the form of fish, which they use. But given that, I feel the population is pretty much in par with what you have on the mainland, except for the Sri Lanka. So that's for the genetic variability, because we had these for the stephensii. We also looked into the gene flow, which is measured in terms of these FSTs, the expected heterozygosity and average expected heterozygosity. This is a pairwise comparison across all the population. So for example, Sri Lanka and Bangalore show a very, so it goes between 0 to 1. The closer to it to 0, there is a less differentiation. If it is 1, it is a very kind of separation basically happening here. And you can tell some of the populations are much closely than some of the other. When you put it into the perspective of the geographic distance here, what happens now? The top one is for the all population, and the one and the below is only with respect to the island populations. As the distance is increasing, the separation, the differentiation is also increasing in all the population, but that also stands for the island population as well. And now that makes me very surprising, that which also comes to the point that habitat probably for Stephensii is so segregated, the population are not mixing, right? They are kind of there, basically. I haven't done this for albopictus, but I bet this will be very different when we run this kind of image for the albopictus there. We did the same PCA plots for these populations as well. This is also to understand how segregated or how much genetic cluster or mixing is happening in these populations. And you can see there are actually 1, 2, 3, 4, 5, 6. This is, please don't take it very seriously. This data, this analysis might change, but this is just to give you an idea. So based on here, what it shows that Sri Lanka kind of stands on its own as a separate cluster. It's not mixing with any. So Sri Lanka, based on this analysis, is not colonized from India because none of the genetic cluster is mixing with any of the Lakshadweep or India mainland population. But what you do see that Mangalore and Lakshadweep, they do share a lot of mixing here. And Mangalore is the nearest port from the thing. Do you know what Mangalore? Mangalore also has the highest malaria API in southern India. If you remove malaria from there, you can actually will say South India is malaria free, which also makes you think that if there is so much of mosquito movement is happening, so this population can be a threat going forward if you have enough number of parasite or happening in those populations. So this is just a way of saying that when you have genomes for different mosquito species, you can actually come and understand the invasion kind of biology and what might be going on and infer the movement of these populations in space, as well as you can do it in time. I'm not going to show here, but you can also figure out the effective population size, also when they might have colonized those islands, and there's a whole thing to that. What else can you use to do this analysis or do this information for? So this is to do with the spread and movement, which people talked about, population differentiation we are talking about here. But also just to make the point for the island, you have very structured thing going on for the island, which actually fits with what you were just saying, that populations are quite segregated there. I'm going to switch the gear for a non-model species here, which we talked about in avian. We have so many avian malaria discussion here, where we mentioned that this is with respect to Himalayan sites where I walked long time back. So these small insects, which is called the Culicoides, they are the main vectors for Hemoproteus, which is one of the blood parasites for these birds. But the important thing about Culicoides is this is also a very dominant, very prominent vector for blue tongue virus in India. It's endemic too. So this is why a lot of phylogenies for Culicoides, which is done in India, are with respect to this particular species. There are so many different species. But one of the challenges for the identification, I don't have, is these species are that it's the identification. The taxonomy is incredibly hard. You have to really, first you have to catch them. So these are the traps you put, but identification is based on the spots. You see that they are very different from different species. And I think that's one of the limitations why people, you can create a genome, but you won't know what species I'm talking, and especially if you have such a huge diversity is going on there. So what we did for that, remember, I don't know if you guys remember that I showed earlier that there is a huge altitudinal gradient in the parasite distribution. Say Plasmodium parasites are nearly absent in the high elevation because mosquitoes are not there. Same for the Hemoproteus, but leucocytosome. I wanted to understand that how across the elevation these different Culicoides species are distributed and what's the seasonality looks like in this species. And you can see somewhere here is the peak in the abundance. This is all in the high elevation environment, so July or August, when you see suddenly there are so many Culicoides are there. Now, if you look at from the bird point of view, when birds are breeding in those habitats, that's between April and say end of May end, that's the host breeding period. You have lots of juveniles which are coming into the population, and that's when the emergence of the Culicoides is in those areas. So which means if there is a transmission chances and if those are there, they will be more prone to infection. The transmission can happen basically. That was the whole idea, to understand how the host interactions might be playing some of the roles. And This is the temperature gradient across all different elevation. So what we did, we collected the samples from all these locations from in Himalaya, where we have the parasite data, and we did the first we did the cytochrome oxidase. After identification, we did this phylogeny, and we figured out that the species we have, they fall very separately from them. We don't know, we still haven't named it. You need to name it. You have to describe the species before you can even publish it, basically. This is how difficult it is for a small insect species, a non-model species. Then what we did was, then we also did, we didn't have much money, but we had some samples from each location, and we kind of made a reference kind of a genome, a very draft genome. Because when you sequence it, you need to map it to a species. The closest species, sonorensis, was too far off. So we built the genome, and we had this draft genome, and we went ahead and compared it with the different genome, and this is very comparable to that sonorensis genome versus what we created here. It's a very small genome, it's not a very big. Now, when you look at the structure, so the whole idea was that, do these different populations, are they segregated by elevation? And what we found that they do actually, given the number of samples we have, and it could be an artifact of the data because there are very few samples. There are only 4 samples from each location I'm talking here. So each of these locations has its own genetic structure going on. So they are very well kind of streamlined. And I think one of the reasons I feel why this is happening here, the emergence time from low elevation to high elevation is very different. So for example, end of June, they will start emerging in these 1800 meter. But by the time they come to the 3,200 meter is a very different time, say after a month, because the environment is too cold. So the temperature actually here is driving the emergence of a vector species, and probably that's how they are segregating any movement also. But the fact, you know how these small culicoides, they move? They do swarm, and they like to go around with the cattle and the sheep. And there are a lot of cattle and goats which the local people, they are taking around in these environments. So this is to say that these kind of things you can do for both model and non-model species, as long as you can identify the species and you can describe it, basically. Now, coming to the final part, or the one part of this thing is that, do you know how people use the mosquito control in all around the globe? What has been used for that? Somebody mentioned the, fumigation is there. There is a larval source reduction also, which is done, which is very cumbersome and everything. Somebody mentioned modified, which I'm guessing they're mentioning genetically modified mosquitoes, basically.
I think that modification of mosquitoes using genetic material,
yeah.
I think it is one decade before it was tried, but
in India, right?
Yeah, I don't know
in Delhi,
where it
I think,
is.
yeah. There's a paper on that.
Yeah, it is, why it is not so successful and still it is not implemented.
Yeah.
I wanted to know it, whether.
You wanted to know?
Yeah.
I think, so the thing about, I think releasing any genetically modified organism in the environment, I think you really need to understand the ecology, how that's going to drive, what is going to change. We do not have that kind of information. I'll just show you a few things. I am not going to details. There is a lot of literature. If you guys want, I can pass it on. You can read about it. The fact that we did all those surveys in Lakshadweep to understand how the species richness, how the distribution was actually to put into perspective what drives the, you say malaria or dengue cases. So tomorrow, say you want to use some technology, I think that information would be very useful. Without having any data, how species coexist, and I think it's just impossible. But one cannot ignore that, basically. So very recently, people have used different kind of techniques for mosquito. There's this sterile insect technique where they kind of do sterilization of these insects. There's also Wolbachia is also used. Do you guys know about the Wolbachia? Is that a yes? Okay, tell me.
Wolbachia is an endosymbiont, which is present in most of the insects, I guess. It's not or I'm not sure, but there are different strains, I think, that are found in
Yes.
mosquitoes.
Yeah.
So there is some incompatibility between, if the strains do not match, maybe, I think, between male and female. So then if they mate, the offsprings are sterile.
They don't hatch.
They don't
basically.
hatch, yeah.
So that's correct. So a lot of places actually have used Wolbachia as one of the techniques where you can release the, the Wolbachia infected mosquito, and when they breed, the eggs basically don't hatch. So it actually used as both as a population suppression strategy, where actually it kills the the eggs basically, or as a replacement where you just replace the whole population. Where does genomics come into that? Would you like to know if you're releasing certain mosquitoes, say you have all the information that how the genetic diversity is changing in that population? Is your experiment really actually working also or not? Would it help you to know the genomes of different areas where you are doing this work? Sorry?
Is working or not, for that we need genetic data to see the...
So pre and post kind of thing will help, right? To understand, basically, yeah. And this has been used in, Wolbachia technology was used in Australia, actually, which led to quite a bit reduction in the dengue. But also very recently, there's an article came 10 days ago in the Nature briefing. They're going to use it in Hawaii as well. For the Hawaiian birds' last hope to save them from malaria. Basically, they're planning to release in one of the islands for that. And they have mapped, they have so much long-term data in order to do that. So I just wanted to mention this thing. But apart from Wolbachia, one of the other techniques which has been used, which was used in Brazil, also is this release of insect carrying a dominant lethal gene. So which means is when it breeds with the wild population, then it leads to they don't reach to a functional adulthood. There's a huge big paper based on that. I think you guys should read it. It's very, it got mixed kind of reviews, but it's a very interesting study. I find it very fascinating that how they show how the pre-release the population, how they selected the site and everything, and what happens in the different genomes of the population. It's a very interesting study which came out. And Jeffrey Powell also have lots of other studies which has been done. Brazil also had done one Wolbachia before this also, which didn't work because they didn't have enough ground data to do this. So for example, to release any mosquito species, you can't just decide that I'm going to release in Bangalore. You need to know what's the, say, trap rate, what kind of adult population density are you looking at, what temperature, all kinds of factors come into play. But I find this paper very fascinating. One more thing which genomes can help is, yes?
So do we know which technique is preferred given what kind of...
It all depends who is willing to take what and what kind of money you have. And because I heard, for example, suppression is very expensive than replacement strategy. What kind of temperature, what environment you have, it all. Like I was in Singapore a few weeks ago and they said they used the whole suppression technique because they had very fine scale data and they did the Wolbachia release there, which led to, I think, 70% decrease in the dengue cases. So it's all very context, the geography specific, basically. Yeah. But the genetically modified one has not been used anywhere, the CRISPR-Cas one, which you are mentioning that earlier. Yeah.
So what is actually the principal strategy? What is the principal strategy to think over to release these genetically modified insects using what kind of genes?
I'm not releasing genetically, I'm not even talking about it.
So it is already done because you are working in that area. I'm asking what is the principal genes? So that is most targeted genes. which keeps the host stable. Meantime, it has to make the offspring sterile.
So in the genetically modified, also the principle applies in the same fashion, right? They use this CRISPR-Cas9 where they edit the gene of that section, which basically lead to either population suppression or replacement. The same thing happens there also. It's not kind of just doing one or the other. That kind of this. I don't know the details of that. Yeah. I think there are other people in my
No, just
group
because you are in that area, I'm asking.
Yeah. If you had asked me I would have remembered it. Yes.
What is the tail risk of releasing this mosquito? Tail risk. I mean, rare events but huge consequence.
You mean Wolbachia?
Yeah.
I think there is no risk. You won't see a change actually happening in the dengue. The main
There'll
thing,
be some negative effect.
so the one thing can be happening. So this is based on my interactions with people in Singapore when I was there. So you're not releasing 10 mosquito or 20 mosquito. You're releasing certain number of mosquito who are carrying these genes or Wolbachia. So when you're releasing, of course, they are coming more in contact. It could be more nuisance factor for people, basically. By the time you see that effect, that something has declined, that takes few weeks, basically. And this release of mosquito is done on a sometimes daily or a weekly basis, certain number. There's a whole modeling done based on that. What was the rate of bite rate was there earlier, or catching rate versus when they are releasing, and all of that. All of that needs to be decided based on all the environmental modeling and everything. So besides that it can cause, I don't think, because they're not causing any disease or anything as such.
In India, if you want to do something like this, like a who would be authority to look at the biorisk and the security and all of that?
So I think VCRC, the ICMR VCRC already have a program on Wolbachia. They are rearing lots of mosquitoes, and I'm not sure the details, but that's one of the things they are actually interested in doing.
What fine scale ground data do you need to?
That I don't know. I'm assuming if they are doing all of that, they must be collecting, they must have some site or selection for that, some trial place or something. So the way it is done, you don't go and release it. You need to do some very big cage trials. You see in that, before you actually, semi-wild or kind of, before you actually go out and do it, basically, yeah.
So one thing, logic idea.
Yes.
That can we target male or female? So because female is very important.
Yeah.
Control the offspring.
Yeah.
So can we select a female?
So they are putting in males here. Yeah.
If we target that chromosome, which must not affect the male, it needs to be required.
True.
So we want to increase one side the male, and on the other side
the female.
Suppress the female. Where in both the ways.
So if you read this paper, this will tell you that when they release that mosquitoes with the lethal gene, the females were not mating with the released male. They had preference for that. Something they have mentioned there. Yeah. But then that whole thing has gone kind of in a different way. Okay, I'm just going to move forward a little bit. So the other thing which, for the vector control, which has been used, like you mentioned, the fumigation, insecticide. And insecticide resistance has been a big issue, right? Everywhere you go, doesn't matter what chemical and whatever people are using, it is kind of not working. So there are studies done where people have looked into when you have the full genome of a species, you can look at it, like me, I'm interested in looking at understanding gene flow, population structure, or you can actually narrow it down to those sections. Like in this case, there is a voltage-gated sodium channel gene. This gene is actually functions, it's responsible for binding target of pyrethroid insecticides. So when you are actually looking into, say, several genomes for a mosquito in different populations, you can actually figure out that is there any selection kind of going on? Will it work? So this particular study, which I have mentioned, they did it on Aedes mosquitoes. What they show here that when you look at the population structure across the whole genome wide, it shows you that different populations are structured in a way. Unless you narrow it down to this VGSC gene, they all fall in the same line. What they are saying is that most likely the invasion happened from, say, similar identity by descent, from the same location. That's why they kind of have same resistance pattern, which actually helps them to understand what should they be changing on the ground if they were to change the way they are using insecticides on the ground. You don't need to do whole genome sequences. There are PCRs which you can do, figure out whether there is resistance or not. And if they did, I think only on 80 mosquito or something to understand this kind of scenario. So there are lots of ways people are looking into genomes, which I'm just giving you a few examples from the control point of view, but it can go way forward than that. We talked about dengue, but I just want to mention that something which is, I feel, is very completely ignored from a mosquito point of view is the blood meals they take in the wild. The fact that some are very specific, some are going and finding different kind of hosts. They are continuously, having this kind of prey-predator kind of relationship, and also transmission of disease. But there are studies which have shown that if you can look for the blood meal, you can find antibodies for different parasite. And getting samples from humans is next to impossible in India. I mean, you cannot do, but that could be one of the source. You can build you will not find which individual, but at a space and time way, you can do these kind of work. And something like that, we were interested in Bangalore, which we are doing it. We did the whole ecology work for the dengue mosquito, where we did how the seasonality of larval habitat works and how that kind of matches with the dengue cases in the city, what drives the larval ecology, but also now looking into the different blood meal scenario, basically what different viruses. Do you know one thing that about dengue mosquito, which I think Kavita mentioned this morning about the transovarial transmission? Do you understand how that works and what implications that might be having on the way dengue is handled in the country? Why we don't have vaccine for dengue so far? Because of that reason. No? So usually, I think what I hear is that, so dengue, you all know they have 4 serotypes, 1, 2, 3, 4. So if you have an infection with the one, next time if you get with 2 or 3, the severity, it's so much higher. It's not like you have a neutralizing antibody which protects you. We know some information from whatever sequence data which Chitra mentioned to you, how many genomes she mentioned, barely less than 1,000 genomes we have. But imagine if there's a transovarial transmission happening because of the mosquitoes can be laying eggs, which means given and you have mosquito, this dengue transmission is also happening throughout the years in a landscape like ours, say in the city, which means what happens to the females if there is a replacement is happening in the population? For example, adults, human adults, or anybody, if it is found in, say, infected with serotype 4 or 3, everybody say, oh, this is serotype 2, very prevalent. But when you are sequencing these mosquito samples, and if you're finding different serotypes, which not being so prevalent in the population, you can actually figure out what kind of, what do you call the divergence or evolutionary terms selection might be happening in the mosquito population, how that might be driving in different locations or different parts of the city. So, say for example, if you do a very fine scale genomics for say dengue mosquito in the city, you know the areas where you have very high prevalence of dengue cases and some areas where have you low. You might say that gene flow might be very higher in the areas where there are a lot of cases happening. That's just my hypothesis, as opposed to the areas where you have less. It could be the habitat, it could be n number of factor. Let's say one of those things, but then you also sequence dengue mosquito is infected with different serotypes, genomes I'm talking about here, and you compare those genomes. I think those information can be very valuable in order to understand that how the replacement, also to predict how that might be affecting that localized population in different places. And I think this is something is completely missing, which nobody is looking at from the mosquito and pathogen point of view in our country, basically. So that's just something I wanted to throw it here, and I think that brings me to the end.
This minimum infection rate, how is it computed?
Which one?
Minimum infection rate.
Oh, so minimum infection rate is basically, so when we catch the adult mosquitoes, you have, say so many traps, these are mentioned, all the dots as you, maybe you cannot read. So basically you have traps all around the, say one area and you are catching say 10 mosquito here, 20 here, whatever. So we don't actually screen single mosquito, you do that into the pools. So you have a pool of say 10 mosquito, 20 mosquito. So that pool divided by the total number of pools you have screened multiplied by whatever, that gives you a kind of infection rate. That's like a standard way of calculating it. Once you do that, you can compare it between different areas. Huh?
The unit is pooled across the entire area.
Only for that area.
Only for this particular area that you're showing.
Say ward-wise. So say if you have it for different wards or whatever your unit is, you can say which area has a high infection rate. How does changes with the season or?
In this August, that's consistent across
that
the
was
wards?
Last year, yeah.
You're using ELISA?
I have not done serology yet. Yeah. No, this is all, no, we are saying that using the blood meal, one can do the serology, not just for these, for any other pathogen. People have done it for JE as well and other things as well.
Yeah, so what would be really cool is to actually look at serology of all of those and how that is changing seasonally over time. And just to say that, the approaches which Cliff talked about yesterday, this multiplex serology is something we are hoping, I don't know, if we ever get that Northeast project, to work with Rahul Roy at IAC to develop. So those where you need, you talked about it, right? 1 microliter you need, that's all you need. So you could actually do multiplex to look at multiple things.
Yeah, and you can tell what blood meal it is, like what the host and everything from that. That's not a problem. Yeah. Also, there also has been a huge shift now that people have been, we also have done some exploratory, but people have been sequencing viruses from mosquitoes, not just the known ones, unknown ones, like what are the insect-specific viruses, how they change with the habitat and everything. So, for example, there was a study recently done, they saw the correlation of, I forgot the main detail, they found some of the viruses which the mosquitoes have in that population, they did it in the lab. It confer resistance to dengue infection. So which means, will that mean, I don't know, you need to test it in the wild, that if you have some of the mosquitoes, say Aedes in the population, if they have higher prevalence of those viruses, does that mean those areas will have less dengue infections or something like that, some kind of thing going on? I don't know. But I think these are, from a pathogen point of view, from genomics, the power, there's a huge power. I mean, you can do so much analyses and understand, which layered with the ecology at the ground level will give you a very fine scale data and help you to understand what might be happening.
I also had one point, so another thing, we're talking about population dynamics. One thing which would be really cool, I don't know whether you guys have done this, but with vector genomes, like different species. For example, if you say one species more of a generalist, or one species seems to have recently invaded, and so on, we can use this genomic data to also look at population history. That is, how has the population size of that species changed in that area? Okay, so how do we know it's in that area? Well, we would have to probably compare it to another area. You want?
Namrata is doing it.
Okay, great. And there are very recent approaches called GONE, for example, where you can actually look at very recent changes in population size over the last 20 generations. Okay, so very, very recent changes. People have been using this for endangered species, which still makes them go back 100+ years. But in the case of mosquitoes, that co-dynamics of different species would be really interesting to test. Sorry.
And I also want to say that considering we had so much malaria and dengue in our country, there were very few studies which are done using even microsatellite data. They're very sporadic, only few on mitochondrial DNA, few with microsatellite, none using the genome. I think probably ours would be the first one which will be done. Yeah. Any other question?
I just wanted to say this is, in some sense, like what you saw from Cliff yesterday, where they're looking at serology of Nipah in the bats, they're looking for Nipah in the bats, and they're looking at infection rates, right? here, this is almost, it's kind of similar, right? Where you're looking at presence of dengue in the mosquitoes, serology positivity of dengue in the mosquitoes, and also maybe infection rates, correlating it with infection
Yeah,
rates.
so I mean, imagine that there are not many dengue cases reported from that area, but you're finding a lot of mosquito positive with dengue. What does that mean? Are those cases undetected or what, basically?
It's just clarification.
Yeah.
So while trapping mosquitoes, you are trapping in large. So say for example, 100 mosquito, if you carry it to your lab for further processing,
Yeah.
how do you make sure that all of them belongs to the same group?
Yeah, we
How do you
identify them.
segregate male, female, or
Yeah. We do the
among the species? Just morphological?
No mosquito goes under the vial unless they are identified first. Yeah.
How do you identify?
We are ticks is specialist in mosquito. That's all I would say. We have a fantastic insectary. If anybody wants to visit, I'll be happy to take you. We have one floor just full of mosquito lines. We have so many lines of Stephensii. Oh yeah, one thing I forgot to mention actually. So Anopheles stephensii, like Gambi, also have 3 ecoforms. One is intermediate, one is Mysorensis, and one is the type form. So far, nobody has sequenced them to understand. Morphologically, you cannot tell them apart. They are found in different niches, just like Gambi. But also, the only way when they lay egg, you can tell just by the egg ridges. So now we have all the 3 forms in our insectary, and we have sequence. I'm hoping we'll do this analysis. So we'll see, do you have those kind of thing, inversions? Is that the reason why they're not mixing and what might be driving? Or, most importantly, the colonization which is happening in Africa, is it by one form or is it by anything which might? Because in Iran, when they had outbreaks, it's usually by Mysorensis. But a lot of what we catch here is the type form, basically, here in India. Basically. So those things are interesting, and I think from an epidemiology point of view, what competes and outcompetes the other ones, basically. Yeah.
Is there a way to
Track.
track mosquitoes?
So people follow the migration, swarming behavior, that kind of thing they do. But not all mosquitoes have that swarming behavior. But in Africa, if you read the literature, they have been following the Gambi and everything, how they are migrating in the night and been catching. Guha was telling me his supervisor used to throw a balloon to catch them with a sticky balloon and things like that. Yeah. But a lot of work which people do on mosquito is all based on the locations where the entry points can be, like seaports and airports and things, especially on the islands, basically, yeah, where they have real worry about something new coming, yellow fever mosquitoes. Okay, everybody done? Thank you.